All right, everyone. So this week, a little bit of a change up of what we typically talk about. A lot of what we talk about in my genetics courses are more molecular, and today we're going to be talking about chromosomes and chromosome abnormalities. So we're going to see some more physical manifestations. I have a couple pictures. I didn't pick anything too disturbing, but I have a couple pictures that I don't know, of like actual people that have a genetic disorder. So if that kind of bothers you, then maybe check out the slides beforehand. But so you're just not surprised. But I didn't pick anything too crazy because there are some kind of disturbing pictures of problems that can happen. But it's important to understand what's going on. So first off, I apologize for the quality of this picture. It's like the best way that I've seen like to show how to actually look at a chromosome. So we have our chromosomes, right? We've talked about that before. Something I haven't emphasized too much, I think I mentioned it before, is that there's a short arm and a long arm of your chromosomes, okay? Now, in this case, we have a single chromosome, so it has not undergone the S phase, okay? So we just have one sitting there, and we're going to kind of keep track of these. We're going to be approaching these from a human perspective. Of course, other creatures have variable numbers of chromosomes. There's some plants that have like hundreds. There's lots of creatures that have less chromosomes than us, more chromosomes than us. It doesn't really matter, but we're focusing on humans. And basically, we're going to see some of the nomenclature that's going to come up, and I wanted to make sure that we know what's going on. So there's your chromosome. In this case, we're looking at chromosome number 7. So you would say there's chromosome 7, and then you say which arm it's on. So is it on the short arm, which is the P arm, or the long arm, which is the Q arm? And then we actually categorize them by bands. So this was before we actually had fully sequenced things, so we could look at chromosomes long before we had the ability to sequence. And so we just kind of organized them this way. And so you would do a karyotype and you look at chromosomes and you'd say, okay, well, we know that if we get rid of this little piece here, there's a gene in there that's important. So we know that it's on the Q arm, and there are different bands. And so it's the density of the chromatin that shows up. So when you see like dark areas, it's more dense, light areas, it's less dense. And so we can band them, so we name those bands. And those have different regions, and so you say, okay, region 3, then there's a band here, and then a subband there, and that's kind of an older school way of actually saying where a gene is. Now we can say like, oh, it's at this region within the chromosome, but we still actually see this kind of location nomenclature in real life. And if you're talking about chromosome abnormalities, then a lot of times you will actually see this type of nomenclature, which we're going to see a bunch of these when we get to like inversions or deletions and things like that. So that's what we're seeing there. Now, there are kind of two categories that I'm going to tackle here with the types of abnormalities that we see. Now, the numerical abnormalities are the ones that you're probably going to typically think of. So that's like your Down syndrome, your trisomies, your Klinefelter syndromes. And so we're going to talk about those first. We call those numerical abnormalities. So you're just taking either having extra chromosomes, like all of them, you have an extra copy, or maybe only one of them, you have an extra copy of the entire chromosome. Now, there are structural abnormalities as well, and those are more like if we delete a portion, or if we add a portion, or if we swap portions between chromosomes. So it's not the whole chromosome, but it's just a sub piece of that, and that's what we're going to call structural abnormalities. Now, if you want to dig a little bit deeper, this is a really interesting paper. I have the link down below there that talks about, they were just studying, they did like for 2 months, we're going to keep track of when we do amniocentesis, which is basically you just look at the baby as it's developing during pregnancy, and you see, okay, what kind of issues do we actually encounter? And then they just kept track of that. And so I'm kind of basing the whole order of this presentation on this. So because they actually addressed like all of the things that we wanted to talk about, including some others that I hadn't addressed before. So we're just going to use that as our little jumping off point here. Get into it. We're going to start off with triploidy. Okay, so basically what happens is, let's say you have an egg, you're a female and you have an egg. You're only supposed to have one copy of each chromosome in there, right? Because when we separate them out, we separate our chromosomes, our sister chromatids, they get separated out. Your gametes should each have only one copy, and then you match that up with like a sperm that only has one copy. So you have one copy from mom, one copy from dad, and you make a baby. Wow, awesome. What happens if you have 2 copies of each chromosome in there? Well, that's not good. Now you're going to have 3 copies when you combine it with that sperm, right? Because you get 1 from dad and 2 from mom. Okay, or you could also have one proper ovum, okay, so that it has 1 chromosome, and then 2 sperm could get in there. We call that polyspermy. So those are the ways that you could accidentally get 3 copies of each chromosome into a single cell. It's relatively rare. You're probably going to early terminate. If the baby does come to term, it's probably going to die within a day or a couple days. Now, you can find case reports for all of these things where maybe they survived, like the tetraploidy example I have, the kid survived till like 1.5 years, but their quality of life is incredibly low. They suffer greatly from cognitive problems, physical deformities. It's kind of crazy. And basically what's happening is you have too many genes, right? You're making these proteins, you're making too many proteins, your cells can't handle it. It's just, you get these crazy things happening and the cells just don't work well. Now, a lot of times what we think about is two gametes coming together and causing this problem. Now, you can have this occur after fertilization. So we call that mosaicism. And basically what's happening is one of the early cells is where this happens in. So you're supposed to line up along the metaphase plate, right? And you're supposed to pull apart during anaphase each of those chromatids. Well, what happens if you don't line up properly? Well, if that happens early on, one of those early cells could have that problem. And so one of the daughter cells ends up with like all the chromosomes, and the other daughter cell ends up with none. And so that one usually just doesn't survive because there's no DNA anyway, so it goes away. And so you're left with this cell. And so depending on how early that happens, all the subsequent cells, so this would be stem cells, so all the subsequent cells that develop from this cell are then going to have this problem. And so that's where you can have like triploidy mosaicism or tetraploidy mosaicism. Now, a lot of times or other things, so you could have mosaicism of any kind of chromosomal duplication, so you can have mosaicism. Like you could have Down syndrome mosaicism, where not all of the cells in the person are actually affected, but honestly, this usually happens pretty early on, and so you're going to end up with some kind of physical abnormality.
Cheeseburger! Say, I love cheeseburgers! Say, I love cheeseburgers!
More often, these children can come to term, but they still don't have good quality of life. They still suffer from a lot of the same problems. If you have triploidy, so that's 3 copies, you can also have tetraploidy. And so in this case, if you want to read this article down here, you can follow the link. That is a case study of this child. Specifically, they had tetraploidy and they were able to bring it to term. So if it has poor survival. I mean, this kid, he could barely breathe. They said most of the problems he was suffering from poor muscles, couldn't really do much of anything, and he was suffering from a lot of lower respiratory tract infections. And so not a great quality of life. This is at 1.5 years, which is very, very rare. They typically don't survive that long. I think this article actually, they mentioned all the other case studies that they could find, and there were like 10 total. But basically, during mitosis, they aren't lining up properly. They're not getting separated properly. And so you end up with all the chromosomes not being separated. So they all stay in the same cell. Or you can just straight up skip telophase and cytokinesis. So you're ending up with the same problem. You end up with one cell. You didn't make the daughter cells. And so it has all of the chromosomes. And that can happen in a couple different ways. Either you're not separating them or you're just skipping cytokinesis. But it can happen early on. You could they talk about hypothetically you could have a diploid ovum with a diploid sperm. That is hypothetically possible. The odds of that happening are incredibly rare. So they think it's actually usually like early on you get this happening and then you result in like basically they're usually mosaics, but I mean most of their cells are tetraploid. You can get a lot of this happening in cancers. So we actually typically see that because you're going to spontaneously abort most children this way. But you can have a lot of cancers that end up with triploidy or tetraploidy, and that can contribute to the cancerous ability of those cancers. That's a weird way to say it. But basically you can, a lot of cancers will exhibit these problems. And basically it allows them to overcome a lot of the checkpoints that we have. Which is kind of interesting. Get to some of the less rare examples, still relatively rare, but they're less rare. So trisomy, if tetraploid and tetraploid are when you have 3 copies of every chromosome or 4 copies of every chromosome. Trisomy is when you have an extra copy of one chromosome, okay? And so it's hence triploidy of one chromosome. Now, the incidence increases with maternal age. That's the strongest correlate, but it can also increase with paternal age if the mom is over 35. So there's some like double correlation if the dad is also older, which can explain why there's such drastic increased risk. You see the drop of risk here more because, so this is age and then the risk as a percentage of having a Down syndrome child. Or any kind of trisomy child. We just usually refer to this as being Down syndrome. You can have basically a trisomy of any chromosome. There's nothing special about Down syndrome except that trisomy 21, which is what Down syndrome is, tends to result in offspring that can survive, and hence, so there's a sort of a survivorship bias towards Down syndrome. But you can see this drops because females become less fecund over time. So anyway, so there's three big ones where you can have some survival. So Down syndrome is trisomy 21, so you have an extra 21st chromosome. I think that's what I have down here. Well, the quality of that is really low. It's lower than I thought it would be. So you can see there's 3 copies, 1, 2, 3. A lot of karyotypes just have really low quality. I don't know why. So 1, 2, 3 copies of chromosome 21. Looks small on this picture, but it's going to have tons of physical manifestations and developmental defects or deficits. Yeah, I mean, we've all encountered someone with Down syndrome at some point. At least most people have. 1 out of 5,000 births. It's still relatively rare, but most of those people with, if you have early intervention, that's the key. For any kind of genetic disorder, we have friends with kids that have genetic disorders I'm going to talk about. The key to any kind of disorder or any kind of cognitive issues is early intervention. So as you grow up and you have your own children, early intervention is the key. And some of the more successful school programs will have really early intervention. It turns out if your kids got issues, be they from a chromosome abnormality or just some kind of mental issue, early intervention, that is the key. So Down syndrome we encounter, trisomy 18 is called Edwards syndrome. We had one of my wife's best friends lost their first baby to Edwards, and so it has an extra copy of the 18th chromosome. So only 5% of babies actually come to full term. So she lost her baby, I want to say 5 or 6 months, and only a 10% 1-year survival. And so most of the time, that's the same for Patau syndrome as well. Lots of problems, lots of neural tube defects. So that's where like basically your brain isn't developing properly, if at all. So microcephaly often, you're typically going to just spontaneously abort. Lots of physical deformities. A lot of these have strange deformities of the face, especially Patau. So Edwards will have really massive cleft palates, right? So basically like your lips don't reform properly at the top of the lip. So basically like the center of the face is what's getting built last, and that doesn't get built properly. Patau syndrome that one, a lot of those will have like they'll only have one eye, or like the eyes are developing and they'll only end up with like one eye in the center of their face. And so you can look that up. I didn't put a picture because I don't know, some of them can be kind of disturbing. But lots of physical deformations of the face. I mean, they have other physical deformations as well, but lots of neural tube defects and a lot of heart defects as well. So basically they're genetically, they're just not proper. They're kind of messed up. And so, not that the kids are messed up themselves, but like their genetics are just screwed up. And so it results in a lot of these problems. And so there are examples of kids living past this. I mean, if you go to the Wikipedia page for, I think, Patau or Edwards, there's like a 14-year-old. And so they can survive, but their quality of life is really, really low. Now, if this occurs in a sex chromosome, okay, so if you have trisomy of a sex chromosome, that's what I want to talk about now. So, for example, I went to a church that we attended in Colorado. I worked on the worship team with a guy that had Klinefelters. I say a guy because he did have a Y chromosome, but he just had an extra X chromosome. You can have extreme Klinefelters. So extreme Klinefelters would have a third X chromosome. Basically, you exhibit female characteristics. This is assuming that you don't have early intervention. You have female characteristics, but male genitals. But you're going to suffer from, like, the testicles will be small, but basically almost everything else is female. So they'll develop at breasts. I mean, this only says 30% of the cases, but it's tricky because a lot of times you'll have early intervention. Usually you give early testosterone and you can reverse most of the physical defects, but you will be sterile for the most part. And so that's just what happens. So that's would be, you can imagine you're supposed to get one X from mom and one Y from dad, but what if both of those X's came from mom? Or you could have like a sperm that has an X and a Y, and that got into the cell as well. And so that's how that could occur. Now, another example would be what happens if you get the X from dad, let's say, or from mom, it doesn't matter, and then from the opposite parent, you don't get either one. So we call that Turner syndrome. So you have one X chromosome and no extra X or no Y. So they're female for the most part. So most of their physical attributes are female, but you have a bunch of body abnormalities. Just kind of depends. Variable levels of mental disability, typically based on early intervention. They're generally sterile, though so one of my wife's best friends from Colorado, this is another one of my wife's best friends, their first daughter has Turner syndrome. And so they've had lots of early interventions. I mean, this kid has had like 15, 20 surgeries, like heart problems, lung problems, all kinds of crazy stuff. She doesn't physically look that different. Her face is a little bit different. But other than that, you wouldn't really know. And I mean, her mom's a special ed teacher, so she's got like crazy early interventions. And so, with early intervention, you can generally have a normal life. They're overall sterile, but they can result, I think the number was like 2% can bring a child to term. But for the most part, you're going to be the gametes that you generate can have these problems. Like you might not be sending an extra, you're not sending, half your gametes are only going to have one X chromosome, the other half aren't going to have any X chromosome. So those offspring are not going to be viable for the most part. Now, another issue that can happen is called supernumerary Y or double Y syndrome. I think the older school term for it is supernumerary, but double Y is more commonly used. And basically, you're a dude, okay, so you're male, but you have an extra Y chromosome. For the most part, not really an issue. It doesn't really cause that many problems, which is interesting because if you have like XXY, you have the extra X chromosome, that can kind of mess with you. That's Klinefelter's. But if you have the extra Y, honestly, you just tend to be taller. That seems to be the biggest issue. But you can have an increased risk for learning disabilities, but for the most part, they're relatively normal other than having an extra Y chromosome. So that's supernumerary Y. You could also have triple X syndrome. So clearly it's going to be female. There's no Y chromosome, so you can't basically generate a male phenotype. But you are female and you also have increased stature and also the potential for learning disabilities and infertility. But for the most part, it's kind of like supernumerary Y, where like it's a female with an extra chromosome, and overall, like they just kind of have relatively mild phenotypes. But it just depends, it depends on a variety of factors, probably environmental and early intervention. And so this came from a paper that was talking about just the variety that you can see in females that have triple X. So that, for the most part, is our numerical abnormalities. There are other numerical abnormalities, and there's some subdivisions within there, but for the most part, that's what we're going to cover in this class. Now we're going to move on to structural abnormalities. So this is not a full chromosome being added or taken away. This would be taking chunks of chromosomes and deleting them, duplicating them, inverting them, doing all kinds of stuff. So we're going to talk about those. We're basically going to follow down this list. This is a chart just to kind of show you what's going on, but we're going to go through each of these. So let's get to it. So the first one's translocation. So translocations are where you take a chunk of one chromosome and swap it with another chromosome. So you're not deleting it, you're just swapping two pieces of chromosome. So in this case, we've got chromosome 4 being swapped with a piece of chromosome 20, and you're just swapping a chunk of them. That can be reciprocal. Okay, so that's typically what we would think about. Reciprocal means that you're just swapping two chunks. So this would be a reciprocal one. If you perform that exchange, that translocation, and it basically happens near the centromere, so where you're cutting a piece near the centromere, we call that a Robertsonian translocation. It's almost identical. It's just you're basically swapping like the little head of this chromosome with the tail of the other one, or if that's just kind of how I look at these things. Um, but it's really not that different. You're just if it happens near the centromere, we call it that. For the most part, translocations, because you haven't lost or gained any genetic information, you've just kind of moved where it is. For the most part, they're silent in that you just wouldn't know unless you actually did a genotype. Now, what you can have some for infertility because your offspring that you generate might have some more issues, and so it can kind of compound, which we see that a lot with a lot of genetic disorders like this. But for the most part, yeah, you wouldn't even note it. You It will not cause cancer, but there are a bunch of cancers that have reciprocal or Robertsonian translocations. In fact, if you go to the Wikipedia article for it, you can see a whole massive list of them. Like this one, I think is cyto something myelogous leukemia, I think, is from a paper. And they're just kind of pointing out where the translocation occurred between chromosome 9 and chromosome 22. You can't see the white part. They just didn't do a very good job with it. But there's actually a couple translocations here. So you'll see some of the nomenclature that I'm using. So if they typically use a nomenclature, so in this case you put a T, that means translocation, and then you say, okay, it's going from this chromosome. So in this case it was going from chromosome 9 to chromosome 22, and then you have the location where it occurred. So just kind of interesting. But yeah, translocation. You can have inversions as well. So those are kind of goofy, where you take a chunk of chromosome. So you take the chromosome and you just flip it. So if you do that outside of the centromere, okay, so if it's not including the centromere, that's called a paracentric. If you include the centromere, we call that pericentric. And so if it's paracentric, that means you're cutting one arm in two spots. If you're pericentric, that means you're cutting, each arm in one spot. But it's really not that different. And honestly, unless you're cutting right through a really important gene, which is, it could happen, but it's relatively unlikely, there's not usually any issues. So just like with the translocations, the actual genes have not really been changed. Okay, so for your translocations and your inversions, you tend to not have a lot of issues. In this case, there's a chromosome number 9 inversion, where basically what's happening, it's hard to tell. Like if you look at any of the karyotype pictures, I just gotta take their word for it. I'm like, okay, I guess that looks like it, because these two look the same, but this one, it's like, this one looks like this, but this one's like flipped, I guess. I don't know. I put it on there because there's always pictures of them, but I don't know. I don't really see it in half of them. All right. If you start deleting stuff, that's when the problems can occur. So if you have a deletion, it depends on where the deletion of the chromosome occurs. So if it's at the end, like you cut off the little tip of it, then that would be a terminal one, because it's near the end. It's at the terminal. If it's interstitial, if it happens like somewhere inside, so this would be an interstitial deletion. If you have a really small interstitial deletion, we call that a micro deletion. And most of the time, that's what we're talking about. It's small, tiny, under 5 megabases for your deletions. If you had a terminal or interstitial, a large deletion, I called it a macro deletion, just to call it something, as opposed to a micro deletion, you're going to spontaneously abort. And so you're not going to carry the baby to term. But if you have a micro deletion, sometimes you can carry them to term, hence these two pictures. So there are a variety of deletions that can result in offspring. Like you're basically not deleting enough genes that are too detrimental to the baby. And so you can carry it to term. So there's a couple of different syndromes. There's a variety of them. I just picked two. Wolf-Hirschhorn, it's on the 4th chromosome, in the short arm of the 4th chromosome, which is the P arm. And then you have Williams syndrome, which that one's a really interesting one. So I kind of fell down a rabbit hole of Williams syndrome. So it's a hemizygous, so it's not homozygous. So both chromosomes don't have the deletion. It's only on one of them, and it's on the 7th chromosome in the long arm at this location, so at the 11.23 location. And it's really interesting. They have, it's debated whether or not, they used to call it elf-like syndrome, and so they think actually a lot of drawings from back in the day, like in the 1700s, of elves were actually maybe people that had Williams syndrome, which is interesting. Some people with Williams syndrome don't like being called that, which I totally get that. It's mildly offensive, I guess. But anyway, but their physical manifestations are really interesting. So they'll have typical characteristics, like you can see this young girl right here, but their actual interactions with other humans is really, really interesting. So they have cognitive deficits, low IQ around the 60s, but they will their verbal acuity is above most people, and so they'll use words that most adults would not use in conversation, like advanced words, and they're so it's kind of like a manifestation of um, like an autism spectrum. Most of these people that if they survive have they're on the autism spectrum, but it's just really interesting. So the Williams syndrome children or people tend to have they'll it's also argued whether it's an offensive term or not. They call it cocktail party syndrome. And so, like, when they're actually interacting with people, they love talking to people and interacting with people and telling stories and using very, flowery is the wrong word, but like emotional language and just really good language. And they're just really fun to talk to, apparently. And so that's just kind of fun. Why did I put this in there? Because I don't know, it just a lot of these genetic disorders can just be super depressing and they make people that just don't have good quality of life. But the Williams people seem to be super happy and have like a great time, which I don't know, it just made me really happy. Um, you can have duplications as well. So for the most part, I mean, it just depends on what's getting duplicated. Um, just like a deletion, it's the opposite. Okay, so it just depends on where it happens. A lot of times this will occur because of when you have crossing over during meiosis. If you have that happen unevenly, then you could have an extra chunk sort of get stuck into that and it gets duplicated. You can also have replication slippage. So that's when your DNA polymerase is chugging along on it, the opposite strand, and it's going along and along, and it falls off. And so it takes that new strand that it was making and kind of backs up a little bit and then starts again. If that happens, then you can end up with a region that gets duplicated. For the most part, I mean, you're having an extra gene, and so, I mean, we know from like having tetraploidy or triploidy, having extra copies of genes can be really bad because you're making too much of specific proteins. Because in the cell, you're going to get transcription factors that are going to bind to specific regions, to specific genes, and cause them to be made. And if you're making too much of a protein, that could be really, really bad. In this case, one of the more common ones that I encountered was Charcot-Marie-Tooth disease. There are other reasons, there are other causes of this disease, but 70 to 80% of the causes of this, it's a muscular dystrophy. So the neurons are starting to actually degrade. It happens during a duplication on the 17th chromosome, specifically peripheral myelin protein. And so if you have, it turns out if you mutate peripheral myelin protein, you can mutate it, you can delete it, and you can duplicate it, and all of those can result in Charcot-Marie-Tooth disease. And basically, one of the main things that you see is this interesting physical manifestation with the curling of the toes. Like you get this, they look like that, which not fun to walk with. A lot of atrophy, and it's progressive as well. So over time, it gets worse, and you die early. But it's like any muscular dystrophy in that way. Now, moving on to the last couple of these are a little more rare. So insertions, I mean, basically it's a translocation of one chromosome into the other. As far as insertions go, they don't tend to cause a lot of problems. I couldn't even find like good examples of them. But I mean, you're not losing any genes. You're just kind of moving where they are. And so hence why it's not usually causing a big issue, but that it does happen. Now, the last couple of these things are interesting, slightly more rare. So an isochromosome, so that's what happens when, so like you've got your two P arms here for your sister chromatids, and you got your two Q arms here, and either during incorrect lining up during metaphase, so they line up like the wrong way, and then the centromeres, so you can imagine if you were supposed to line up this way and pull apart this direction and then this direction, well, what if you lined up like both the P arms on this way and both the Q arms are on this side, and then their centromeres sort of like stick together, and so you end up with your two P arms stuck together and your two Q arms stuck together, or you just completely lose one of them. So it just kind of depends on how severe it is. If it happens with an X chromosome, it could cause Turner syndrome, like I talked about before. But that's basically what you end up with. You have like one arm. What else can happen is like let's say you delete the P arm right here, and then you replace it with another Q arm, so you get this really long double Q armed thing. I mean, it's just a massive deletion of the entire P arm, and then a massive duplication of the Q arm. And so it just depends on where it's happening, which chromosome it's happening to. There's one specific syndrome, so this was one that I found called Pallister-Killian syndrome. And so you can see here, you say I for isochromosome of the 12th chromosome of the P arm. Okay, and so you have an extra P arm of the 12th chromosome, and it just results in the physical manifestations that you're seeing here. Basically, I mean, almost all of these problems end up with mental problems, but also you get like interesting patterning of the skin and facial deformities. Now, two more things to go. So, ring chromosomes are where you basically cut the tips off of each of the end, and then you religate them together. Now, that typically happens in the telomeric region. So, the telomeres are the ends of your chromosomes. You may have heard of telomerase, which is the enzyme that adds onto those. Basically, your telomeres get cleaved every time you undergo replication. And so if you don't restore those telomeres on the ends, eventually you start cutting into the chromosome, and usually that results in cell death. So it's a way of maintaining the age of the cells so that the cells don't get too old, because the older you get, the more mutations you can incur. And so if this happens, you can have like folding over of them. So I found these fun pictures. Those are kind of fun. They just colored some of these chromosomes, and then they showed the ring structures, which is kind of cool. It's relatively rare. It can happen for any of them, and it just depends on the chromosome. So, I mean, you're usually, a lot of times it's a mental problem and a facial abnormality. But it just depends, you know? What chromosome is it actually forming the ring structure? Basically, you're going to transcribe these improperly, and your offspring are going to be messed up too if you're trying to mate with chromosomes that are like this. So they're probably infertile as well. The last thing I want to talk about are marker chromosomes. So if you encounter a marker chromosome, so in this case down here, it doesn't necessarily belong with the 20th chromosome. They just kind of stuck it here because there was space. But a marker chromosome is just a small piece of a chromosome. So you can call that partial supernumerary. So I had supernumerary Y syndrome, right, where you have an extra Y chromosome. Well, what if you have just like a little tiny chunk of a chromosome? Who knows what genes are actually on that? And so that's what affects the severity. So like you could have a marker chromosome of a gene that doesn't really affect much. Or you could have a region that has something really powerful that's really going to cause a lot of problems. And so it just kind of depends. And so that's the last slide I have. So that's just your chromosome abnormalities. Lots of severe issues can happen. Some of them don't have any effect on humans. It just kind of depends.